Hey, my dear students, listeners, and viewers all over the globe. My name is Dr. Bright. I'm also here this evening to introduce to you or give lecture on one of our lecture series titled Environmental Toxicology. Okay? So next now is the environmental toxicology or toxicological termination. So we want to define a lot of terms. In order for us to understand environmental toxicology very well, we need to go to definition of terms using under toxicological discipline. So one is toxicology. Toxicology is the study of adverse effects of chemicals or physical agents on living organisms. A toxicologist is a scientist that determines the harmful effect of agents and the cellular, biochemical, and molecular mechanisms responsible for their effects. So toxicology is the scientists that carry out toxicology. Okay. Here also we have a lot of terms. One is toxins. It refers to toxic substances that are produced by biological systems such as plants, animals, fungi, or bacteria, e.g. zearalenone produced by a mold, which is a toxin. Toxicant is also used in speaking of toxic substances that are produced by or are a byproduct of anthropogenic, that is human-made activities, e.g. dioxin. You see dioxin 2378, tetrachlorodibenzo-p-dioxin, TCDD, produced during the production or combustion of certain chlorinated organic chemicals. It's also a toxicant or poison, as the case may be. Venom. It is a special category of toxins that must be injected by one organism into what? Another. So when that happens, we call it what? A venom. Toxicity or poisoning is the harmful effect of chemicals on the exposed biological system. Like toxicity or poisoning now. Toxicosis describes the disease state resulted from the exposure to chemicals. Okay, so toxicosis. Okay, the other ones are the toxic agent or substance. A toxic agent is anything that can produce an adverse biological effect. It may be chemical, physical, or biological in form. Toxic agents may be chemical, such as cyanide, physical, such as radiation, biological, such as snake venom. So classification of toxic agents. So toxic agents are classified in a variety of ways depending on the interests and needs of the classifier. So toxic agents can be classified according to target organ, the liver, the kidney, the hematopoietic system, etc. Based on use, pesticide, solvent, food activities, based on source, animal and plant toxin, and based on effects, cancer, mutation, liver injury, etc. Now, what is environmental toxicology? Environmental toxicology is the study of the impact of chemical substances, that is potential and actual pollutant, upon the structure and function of ecological system, including man. Environmental toxicology uses typical species chosen from the ecosystem or laboratory test organism to examine the answers for the chemicals. We can extrapolate from this data for the whole ecosystem. Ecological system is defined as a complex system from the molecular level through individual organisms and community to the ecosystem. Here is a chart that shows us increasing complexity from cell to biosphere, that ecosystem to biosphere, and decreasing precision, okay, from the cell to the biosphere. So as this ecological hierarchy increases, so is the ecological responses also what? Increases, or precision get decreased. Slide shows a description of the interaction of these chemical substances with the site of action. So the interaction of the chemicals with the ecosystem and function needed to be described by environmental toxicology. So as these chemicals are entering the environment, their fate and transport is being described as being biotransformed. They undergo biotransformation. Their form changes. and then they now attack a receptor, the place of effect. From once a place of effect has been, which is the receptor, has been attacked or targeted, then we can a lot of effects on the chemical substance on the ecosystem can be measured or observed. Okay, their physiology and behavior can be described. Biochemical parameters can be described. Population alteration, changes in structure and function too of the community can also be observed. Okay, so this is what happens. So when chemicals sometimes are released into the environment, they either in the water or air or the soil, they have a target site, which is the receptor. Once that enters, then consequences or effects on the ecosystem ensues and can be measured. Okay, here now, because of time, we want to classify environmental toxicity testing. So classification of environmental toxicity testing. So environmental toxicity testing can be classified into the following: 1, duration relative to the lifespan of the organism. 2, the number of species. 3, the type of test organism based on taxonomical rank. 4, ecosystem. 5, exposure scenario. 6, trophic level. 7, measurement of endpoint. And then 8, the aim of testing. So number 1, taking from that number 1, duration relative to the lifespan of the organism, is divided into two. 1, the acute toxicity testing and the chronic toxicity testing. Under the acute toxicity testing, it is a relatively short period of an organismal lifespan, which is 28 to 48 hours, where we can use daphne, fish, rat, birds, then check. But I want to say here also that bacteria, algae, protists, they have a short generation time. Like a very important indicator organism for environmental monitoring is the alivibroficheri, which is used for bioluminescence inhibition. Daphne magna test, okay, which is used for immobilization as an immobilization test according to ISO, International Standard Organization, and Organization for Economic Community Developing Countries. So these are the protocols, okay, or SOPs of these people where Daphne can be used or the heartbeat can be measured. So based on repeated exposures, acute toxicity could be subacute as the exposure to the chemical is for 1 month or less. So when we want to proceed from 48 hours, 72 hours down to maybe less than 1 month or 1 month, such studies are now called what? When we want to do repeated exposure, want to continue it. Such tests are called subacute. Then for chronic toxicity testing, which is the second one in that lifespan, okay, or duration, it is a relatively long period of an organismal lifespan, more than 3 months. So once we do more than 3 months study, we will now say, we'll talk about what? Chronic toxicity testing. Here, sublethal endpoints such as movement, feeding, growth, reproduction, immune, endocrine, developmental dysfunctioning can also result in mortality. Example here is that reproduction inhibition test must include 1, gestation period for females and a significant portion of the time for spermatogenesis of what? Males, not multi-generational. Only 1 to 2 generations are what? Involved. Daphne magna test organisms also can be carried out also. Daphne reproduction test for 21 days based on repeated exposure. Chronic toxicity could become subchronic as the exposure to the chemical is from 1 month to 3 months. So 1 to 3 months is subchronic. More than 3 months is what? Chronic. Less than 1 month is what? Subacute. And few days or weeks or hours is what? Acute. This slide is the number of species used in the test, right? Toxicity testing. So it can be single species toxicity testing or multi-species toxicity testing. So single involve 1 test organism, well-known organism derived from control labs, or multi-species toxicity testing, where 2 or more organisms and their interactions are what? Studied. So we can see in the case of multi-species toxicity testing, further explanation there. So if we are, for instance, we are doing a biotest on competition of 2 bacteria species for 5 days. So we measure competition as a measured property, or microbial predator prey test for 3 to 5 weeks. Here we measure predator prey numbers, or we are doing a microcosm tests for 3 to 10 week study. Here we measure property and number of species, diversity study, respiration and heterotrophic activity studies. Or mesocosm. The mesocosm, for 5 to 6 months. And here the measure property is measuring the number of species, diversities, and element cycles, such as nitrogen, phosphorus, and all the elements using analytical techniques. Okay, you can see this is a picture of microcosm or microcosmal toxicity testing. You can see different setup. You can see the one done on track, on drums, on buckets, plants. You can see the one done with plastics, smaller container plastics, maybe for earthworms, for different organisms. You see the one done on bucket, maybe for fish and the case. So this one is like laboratory control testing. It's called microcosm. So when we carry sample from the field and bring them to the lab to check for their natural event, it's a microcosmal study, okay, intact as it is in the field. But you see, the other one is the mesocosm or mesocosmal toxicity testing. In this mesocosmal toxicity testing, you can see the open field, in the marine, in the maybe oil field rigs, you can see in the agricultural vast land, flowers. So when we have this to say that mesocosm is a larger microcosm, and then a longer monitored study. From that previous study, we see 5 to 6 months. While microcosm is 3 to what? Then you can see that mesocosmal study is a longer period monitor study. Here also more trophic levels are engaged, greater complexity. There is greater complexity, unlike the previous microcosm as well. Also, you see, unlike this one that happened in the lab, the mesocosm occur at what? The field. Here, rainfall, they have been exposed to rainfall, sun, atmosphere, just the natural condition are being studied here, okay, in the open field. So the other classification is, based on the mesocosma 2, in this field study, which is mesocosma, observational and experimental monitoring are carried out, maybe in situ biomonitoring, following an indicator organism existing naturally, what we call passive monitoring, or we can place by assessor into the environment. We'll put an assessor, that is something that can assess, evaluate, or monitor the environment. In this case, we call it what? Active monitoring. So another classification system is the type of test organism based on taxonomic rank. Here, bacteria can be used, algae, fungi, plant, animal, multi-species. So, based on taxonomy, you can do toxicity based on this. Carry a selected bacteria, selected algae, fungi, plant, animals, or multi-species studies can be done. You can see in this case, this is a microscopic test organism. In the case of bacteria, as I said initially, the alivibro ficheri used for bioluminescence inhibition. You can see the organism bioluminescence. So this bioluminescence level is what we use to measure toxicity. You can see the algae, the Pseudochyneria subcapitata is being used, the Chlorella vulgaris, the Senadesmon subsicatus, the Protozoa tetrahymena pyriformis. They are all used. This is based on the taxonomic ranking. You can see the plant, the limna minor, which is the common duckweed, the Cinna apsis, the white mustard seed, the Lepidium sativum, the garden cress. So you can see the downward this slide there. You can see these synapses are by white mustard seed exposed to increasing concentration of metal. To the right, you can see growth at the left side, but towards the right, there is less growth because of the increasing metal concentration. Okay. Based on animal, we see the Daphna magna, which is a white flea, the Thamnocephalus platirus, the heterocycles incongruence, and then the Forsomia candida, which is commonly called columbola. The columbola, it just behave like earthworm there. So these are the animal group which we can monitor based on taxonomical rank. You can see bacteria, algae, fungi, plant, animals. So these are representative of bacteria, protozoa, algae, plants, and animals. So based on the tested, another classification is based on tested ecosystem. And the ecosystem, we can look at aquatic or terrestrial. In the aquatic, we look at the water sample or the sediment. Aquatic eco-toxicity, which is the pioneer. Here, the aquatic divided into three: the marine ecosystem, brackish ecosystem, and the freshwater ecosystem. The terrestrial ecosystem is the terrestrial eco-toxicity. Next classification is exposure scenario. Here, the condition of the contact between the toxicant and the body of the test organism. So one of the exposure scenarios is the whole body test. Here, the test organism is immersed into the tested water, sediment, or what? Soil. And here now, direct contact between the pollutant or contaminated environment and the test organism is what? Involved. Here, the skin or all the dermal surfaces, the eye, the gills, the hair, etc. And many cases, the inner mucosal surfaces in the mouth, the trachea and the digestive tract are also monitored. You can see the columella in direct contact with soil. You see here now, here there is direct contact with the soil. You can see in this case the water and sediment contact of heterocypress incongruence, which we call the ostracoda, and the family of the ostracoda. So here they are exposed to water and sediment, but in this columella case, they are being exposed to what? Soil. So this is what we call whole body test. The whole body is exposed to the contaminated environment or the pollutant or the toxicant of study. So here, this one is the one we did in our lab here. You see student intern pass 2015, 016 and 052, 096 and 066, 2015. This student, they took time, cultured this earthworm. They grew it here in the lab, turning from the farm at Amamputo. Got there and then they came to the lab, grew this earthworm, and then they exposed them to different toxicants, battery and the laptop batteries. They exposed them, okay, and they are checking the toxicity, checking mortality, as the case will be. This one we did, the students here did in the lab here, and they recorded their results. Another exposure scenario, apart from whole body, is the feeding studies here. So here, the eating of food or toxicant mixed into food or drinking water. This model the uptake and the effect through the digestive system. The problem here is that the test animal eat much less or not at all when they feel the presence of toxic material in the food. So, or we can also do what we call placement of control amount into the stomach by a tube or what we call gavage, or injection of a control amount intramuscular, intravenous, intraoscular, intraperitoneal, okay, and so on and so forth. So intracranial within the brain, intramuscular within the muscle, intravenous within the vein, okay, intraperitoneal within the lower abdomen, intracranial within the brain, intraoptical within the eye, intranasal within the nose. So they can, in this case now, we can just control amount or dose of the toxicant. So you can see those rats, they are being injected, rats are being injected, either through gavage or through control amount into any of the veins or the sites. Another classification is based on trophic level. The major trophic level we study in toxicity testing is R, producers, consumers, and decomposers, or what we call the detergent. The producers here are the algae, the consumers are the fishes, and even the form of the fungi, like the consumers, the fishes, okay, and some of the bacteria can also be consumers as well. The decomposers, normally the fungi, okay, or the detergents. So we can look at this trophic level, how energy is moved from one point to another trophic level. Another classification is aim of ecological testing. So if we can, based on our aim, we can screen toxicity, screening for toxicity, mutagenicity, that is mutation, teranotoxicity of single chemical based on fetus. The effect on testis is teranogenicity. Okay, screening toxicity, mutagenicity of experimental samples, screening toxicity, mutagenicity and teratogenesis of mixtures, waste materials. Okay, so biomonitoring, integrated monitoring too, early warning system, establishing effect-based environmental quality criteria, direct effect-based decision making. So based on, we can have an aim in eco-toxicological testing. Based on aim, we can carry out any of these ones on using the test chemical samples. Okay, another classification is measurement of what? Endpoints. So we can measure endpoints. Endpoint include the following: growths. Based on growth, when we measure cell number, mass production, root length, chlorophyll content, all these are under what? Growth. Survival, mortality is under endpoint. Immobilization, when there is no movement. The respiration, either by oxygen consumption or CO2 production. Enzymatic activity, so that the hydrogenase, urease, phosphatase and the rest of them. ATP production, reproduction, either sexual or asexual. Luminescence, based on emission of light. So you can see those pictures present. The luminescence is that greenish one below, while the algae is the one on top of the slide. Okay, so we see the columbella there. Okay, based on mortality, survival. Okay, you can see the ATP. You can see the shape of the ATP there in the blue and yellow ink image. You can see the other one, container, something like a valve, aspirator. That one is based on respiration, oxygen and CO2 production. So enzyme activity can be carried out in the soil too, that columbella, phosphatase and the rest of them. So these are many pictures, they depict these measured endpoints. Okay, so these measured endpoints, as major emphasis based on mutagenicity. We can measure the number of mutants, number of revertants, chromosomal aberrations. Okay, under the carcinogenic testing, we can check for tumor teratogenicity, we check for reproductive success, cytogenetic characteristics. Under biodegradation test too, as an endpoint, we check for consumption of oxygen, consumption of substrate, production of endpoint, production of CO2. They are biodegradation endpoints or parameters. Also, other bioaccumulation test, chemical analysis of accumulated substance. Okay, the endpoints, endpoint of measurement and endpoint of evaluation. So endpoint of measurement, all the enlisted biochemical, physiological, behavioral, population, community parameter and ecosystem effects can function as what? Endpoint. Endpoint of the test evaluation, the result of the environmental toxicity testing following statistical evaluation. So the results follows statistical evaluation, typical value for toxicity or other harmful effects for this evaluation and decision making. So the characteristic concentration level can be determined from concentration response of those response curve, as can be seen in subsequent slide. But before we go there, I want to look at what is concentration and dose. What are they? So dose is the actual amount of chemical substance that get into the organism through different exposition scenario. Human toxicology, or amount fed or injected. Okay, concentration is the actual concentration or value or amount, the actual amount or the chemical substance in the environmental element or phase which the organism get in contact with. Response is the amount of damage caused by a specific dose. So types of doses: absorbed dose, exposure dose, administered dose, total dose. Exposure dose is the amount of xenobiotic encountered in the environment. Absorbed dose is the actual amount of exposed dose that enter the body. Administered dose is the quantity administered, usually orally or by injection. Total dose is sum of all the doses. So dose units. So another important aspect is the time over which the dose is administered. This is especially important for exposure for several days or for chronic exposure studies. The commonly used time unit is 1 day, or tox. The usual dosage is milligram per kg per day. Since some xenobiotics are toxic in much smaller quantity than the milligram, smaller fraction of the milligram are used, such as microgram. Other units are shown in the slide below. We can see the kilogram, which is 1000 gram, 10 raised to the power 3. Gram is 1, milligram is 0.001 gram, microgram is 0.000001 gram, nanogram is up to 9, picogram is up to 12, femtogram, as can be seen there, is up to 15. Okay, so it's up to 15, femtogram, 10 raised to the power minus 15. So 10 raised to the power of 12 is pico, 10 raised to the power of 9 is nano, 10 raised to the power of 6 is micro, 10 raised to the power of 3 is milli, and so on and so forth. So in the environmental sciences, environmental exposure, units are expressed as the amount of xenobiotic in a unit of the media. Milligram per liter for liquid, milligram per gram for solid, milligram per cubic meter for what? Air, like that one of the centrifugal sampler. So the unit we measure, if we measure the organism, maybe we say colony per, CFU per milligram per millicube, or CFU per millicube, for CFU, that is CFU, colony forming unit per cubic meter based on air. Smaller units are used as needed, e.g. microgram per mil. Other commonly used dose units for substances in media are parts per million, ppm, parts per billion, ppb, parts per trillion, ppt. The dose level at which a toxic effect is first encountered is known as the threshold dose, which we are going to talk about in the ED and the EC and the rest of them. Doses below the threshold dose are often referred as sub-threshold doses. So the dose level at which a toxic effect is first encountered is what? The threshold what? Dose. ppm. This is a way of expressing very dilute concentration of substance. Parts per million is commonly used as a measure of small levels of pollutants in air, water, body fluid. Parts per million is the mass ratio between pollutant component and the solution. And ppm is defined as parts per billion, is 1 over 10 raised to the power of 9, as I said initially. Parts per trillion is 1 over 10 raised to the power of 12. Parts per million or ppm means out of a million. Usually describe the concentration of something in water or soil. 1 ppm is equivalent to 1 milligram of something per liter of water, or 1 milligram of something per kilogram of soil. 1 mg per kg is equal to 1 part per million. Concentration response relationship in environmental toxicology. Reasons for using concentration and not dose. We cannot control or verify the amount of chemical substance getting into the organism from the environment. So several exposition scenarios like this makes us to use concentration and not what? Dose. You can see this is a picture of that columbella that look like earthworm, where we did whole study exposure scenario, inhalation, digestion of chemical toxicants. So this is a concentration response curve. We said we prefer using concentration than dose anyway. So you can see from here, it is on the right hand, y-axis is the inhibition in percentage, on the x-axis is the log concentration in milligram per kg. So here we're able to derive a dose or concentration threshold. Remember in our previous lecture we said that a dose level, the dose level or concentration level at which a dose effect is first encountered is known as threshold dose. So these are threshold doses: EC10, EC20, EC50, EC90. Their effect concentration causing 10, 20, 50, 90% inhibition in the measured parameter. You can see them: EC10, EC20. You can see the arrows depicting where the effects are seen. So they show responses as well. So endpoint evaluation of endpoint under acute toxic testing, we have the following dose threshold doses, which are EC20, EC50. What does this stand for? If EC20 or 50 stands for effective concentration that has an effect of 20% or 50% on the measured endpoint. So if I'm measuring growth or inhibition, I want to say that there is 20% inhibition or 20% growth. Okay, based on that is the concentration that inhibited or supported 20% growth. ED20 or ED50, these are effective dose that has effect of 20% or 50% on the measured endpoint. IC50 inhibitory concentration that has 20% or 50% inhibition on the measured endpoint. LC20 or LC50 is a lethal concentration that causes mortality or lethality in 20 or 50% of the test organism. LD50 or LD20 is a lethal dose that causes mortality or lethality in 20 or 50% of the test organism. You can see a bell-shaped curve here that shows the additional mortalities plotted against concentrations. The response are controlled by numerous genes like length, coat color, fecundality, and so on. So these are response curve, concentration response curve, which you can call the bell-shaped curve. Okay. So evaluation of results on environmental samples. So how do we evaluate results? So environmental sample where unknown chemical substances in unknown concentration, thusly giving the equivalent concentration with well-known chemicals, chemical substance or elements, e.g. copper equivalent, okay, on alivibrofichery bioluminescence inhibition or 4-chlorophenol equivalents. So here now, in this case, for instance, we try to study with different endpoints: immortality, growth, reproduction, respiration, and the rest of them. So here copper was used as the toxicant. You can see concentration prepared from 0 to 200 microgram per liter. At the end of 96 hours, LC50 was determined as 50 microgram per liter. You can see in the control there was massive growth. In the 13 microliter gram, there is growth. 25, there is growth, but decrease. There was 2 decrease. You can see in 50, 5 died. Out of 10, 5 died. Remaining 5. In 100, 2 were remaining. 8 died. In 200, all died. So you can see, based on LC50, lethal concentration that can kill 50% of the exposed animals, organism, test organism. So 50 microgram actually kill 50%. 50% of that were cleared. So the concentration that kill 50% of the population after 96 hours of exposure is 50 microgram per liter. So you can see this 50 microgram per liter is the threshold concentration. It is the what? The threshold concentration where the toxic effect is first encountered. Okay? So effect concentration are expressed in total or dissolved copper. So this can be extrapolated using Pnex or EQC, expressed in total or dissolved copper. I'm going to express this in subsequent lectures. So here I give a working out a table here. You see the table. This was the result carried out by our students here in our institution. Those students I mentioned to you: 016, 096, 066, 097, and the rest of them. So they use ITEL phone, Gioni phone. So those batteries were ruptured and used at different concentrations, 6.25 to 100, and they use it on leaf of Zime, okay, and different seeds. So here the root length here, they use onion bulb. Yeah, they use onion bulbs. So root length were measured. You can see from concentration to 100. For ITEL, the root length is from 100 to 41. The Gioni, the root length is from 100 to 38. But the inhibition is from 28 to 51. You can see the root inhibition the percentage inhibition is keep increasing. Therefore, Gioni is from 24 to 62. So you see this for here now we have a measured endpoint here, which is percentage roots inhibition, and then we have a threshold concentration which we want to establish here, which is EC50. The effective concentration that we inhibit 50% of the root length of the onion bulb exposed to ITEL and Gioni. So this This was worked out using concentration response curve, using statistical analysis, and then what we call regressional analysis. Regressional analysis under statistical analysis. And that regressional, it brought out a regressional equation, for ITEL it is y is equal to 0.3894x plus 16.48. Y for Gioni, it gives us regressional equation of regressional equation of y equal to 0.5074x plus 18. So here we want to say that the y there is the y-axis value, that is the inhibition. The 0.3894 and 0.5074 are the slope. The x are the concentrations, in this case in percentage for both the Gioni and the ITEL. While the 16.48 and the 18.38 are what? The intercept. So when we do the anti-log, when we find the value of x and then do the anti-logging, we get what? The value, our percentage, our EC50, either for ITEL or Gioni. That is the effective concentration of ITEL or Gioni that can inhibit 50% of the root length of the onion bulb. So this was the study carried about your predecessors, and this was the outcome. And they plotted under statistical software. Okay, they plotted under linear regression analysis. This equation was brought, and they used it to get their EC50 and LC50, as the case may be. Here is another. The spent phone was exposed to Limicolaria flammaria. This one is this organism was exposed at this concentration, 0 to 6 milligram per kg. This Limicolaria flammaria is a snail, the normal giant African giant snail. Okay, to different concentration of this. So we found out that mortality keep increasing as the time of exposure increases. So you can see mortality rates 1, 2, 3 to 13. Mortality percentage keep increasing, and the survivor keep what? Decreasing. And so this percentage mortality was subjected to statistical analysis, linear regressional equation. Okay, so when that was done using the software, the equation y is equal to 9.780x plus 00 was what? Derived. So from this, our EC50 can also be what? Obtained. Maybe I will give you this as an take-home work. We are going to work it out. Maybe subsequent this thing, we're going to work it out. Okay? So I want you to drive EC50 with this equation. How would you get your EC50? So your y is 50, right? So get x 50. Y is your 50. So get x. Make x subject of the formula there. Get your x. The value. Do the anti-log. Do the what? Anti-log, as can be seen in this. Do your anti-log of that value to get your value, to get your LC50 in milligram per kg. So your LC50 should fall between 1 to 6 kg or more. Okay, so this one is take home assignment. Please take home assignment, go and work it out. Your y is your 50, which is your endpoint you want to check here, the concentration that will give 50. Make x subject the formula, get the value of x, find the anti-log of x. That gives you the value is your LC50, letter concentration that can kill 50% of Limicolaria flammaria when exposed to different concentration of when exposed to spelt ITEL phone concentration. So as I said, the concentration can be 1 to 6 milligram per kg or beyond. So go and work it out and get your LC50 there. Thank you. So this is very, very important. Please, you can take notes. Then the evaluation endpoint. So we use all this to evaluate under chronic toxicity testing. We use NOEC, NOEL. They stand for no observed effect concentration or no observed effect level. NOAEC or NOAEL, no observed advanced effect concentration or no observed advanced effect level. LOEC or ACE is equal to /LOEL/LOALE, lowest observed adverse effect concentration on level, MATC, maximum acceptable toxicant concentration. So we want to say that this maximum acceptable toxicant concentration is NOEC plus LOEC divided by 2. So, but we can determine it with ANOVA, analysis of variance, such that concentration of dose that does not or produce a statistically significant effect are compared to negative control. So implicit assumption here is that there is no threshold concentration or dose. Test organism can buffer the effect of the toxicant at certain level. So these are the ways we can evaluate endpoint. Endpoint, especially in chronic toxicity, we can evaluate them using any of these acronyms and more. Okay, so these are models of toxicity of compound at low concentration. So you can see the x-axis is the concentration of dose, while the y-axis is the toxicological effect or enhancement. Either there is inhibition or growth or enhancement. Okay, the thick line is no threshold. The break line is the threshold value or point. The multiple short break lines are the hemotic responses. It can be seen in that chart. Okay, so here now is another classification scheme, toxicity classification of environmental quality criteria. Here we see this table, general criteria for toxicity classification and relationship between LC, LD, EC, and toxicity rating. You can see LD50 in the left hand side, LD50 in milligram per kg, okay, and LC50 in milligram per liter, EC50 of percentage of effluent, okay, and then the last column is toxicity rating. So greater than 5,000, greater than 100 or 100% is, we classify it as what? Relatively not acutely toxicity. 500 to 5,000, 10 to 10, 10 to 100, minor acute toxicity. 50 to 500, 1 to 10, 1 to 10 is moderately acute toxicity. Less than 50, less than 1, less than 1%, very acutely toxicity. So if, for instance, we want to rate here, we want to rate in this case now, okay, we did not do the, okay, like here now is 50 microgram per liter, 50 microgram per liter, okay, so for 50 microgram per liter, okay, for this, how do we rate it? 50 microgram per liter. You see, in this case now, 50 microgram per liter is less than 1. So we say we can classify that copper as what? Very acutely what? Toxic. Very acutely what? Toxic. Then when you come to this table now, this table you can see the EC50 for ITEL is 86.808, while that of Gioni is 65.46. So here now, the 86, 65, when we come to EC50 by percentage, EC50 percentage here, 80, you can see they are between 10 to, both of them are between 10 to what? 100. How do we classify from that result of EC50 in that table? How do we classify how do we now classify what? The ITEL and Gioni spermphone on the root length, inhibition of root length of onion bulb. Okay, we classify it as what? Minor acutely toxic. So with that EC value, we can say it is minor acutely what? Toxic. So we want to say here that EC value is the central, is or the main endpoint evaluation threshold, the main endpoint threshold dose or concentration we use to judge whether something is toxic or not. Okay, so the next slide here shows us acute toxicity units, TUA, which is given as TUA is equal to 100 per liter, or E or IC50, that is LC50, EC50, IC50. Okay, 50. So like here now, the order of that onion bulb here, we have 86 and 86.08 and 65.46. So if I want to get the TU acute toxicity units for those Gioni or item, for instance, what I need to do is to say 100 divided by 86.08 or 100 divided by 65.46. The value now is now what I will now finally classify the toxicity again. The value, if the value is less than 0.4, I say no acute toxicity. If the value is greater than 0.4 but less than 1, I say slightly acute toxicity. If the value I get by dividing 100 by those EC values, if I get my EC values and divide 100, and my value is between 1 to 10, I say they are what? Acutely toxicity. If the value is greater than 10 but less than 100, I say they are what? Highly acutely toxic. If the value after dividing 100 with the EC values I obtain from my calculation, once I divide by 100, okay, and the value I obtain, the final value of my TUA value is greater than 100, what do I report that my toxicant or chemical analysis or toxicity result? I report it as what? Very high acutely toxic. Okay, like if I divide 100 by that 86, I have 1 point. I have 1 point something. If I also divide by that 65.46, I also have 1 point something. So if we are to, based on acute toxicity unit, TUA now, here now we now see that our value is between 1 to what? 1 is greater than 1 but less than 10. How do we report Gioni and ITEL? We say that Gioni and ITEL are what? Acutely toxic. They are acute toxicity. They have acute toxicity. That is our report. Acute toxicity or acutely toxic, as the case may be. Okay? So, the last but not the least there is the risk assessment or scientific basis for establishing ill QC. Okay? So, and for us to do it well, first we need to identify hazard and do data compilation. 3, we need to do exposure assessment that will give us PEC, and then the PN or effect assessment. Either we do exposure assessment, PEC, or effect assessment, which is PNEC, as I had mentioned in previous slide. Then either of the exposure assessments will both result or give us or lead us into risk what assessment. We now say whether the chemical has a risk effect or not. And then also from the effect assessment, we can also do our environmental quality criteria to know. But we want to say, so here we can now see that the environmental quality criteria is can be gotten directly using the effect what assessment. So we want to say here that toxicity, environmental ecotoxicity testing, is a very significant area. Okay, and one organism does not give us a cogent summary or direct assessment. So it is necessary we engage a battery of what? Assay. It's necessary we engage what? A battery of what? Assay. Maybe for a different trophic level or multi-species toxicity testing to give us a wider data or comprehensive data on that toxicant. So using one method or classification assessment is not the best. Then either of those want to also say that 50-point endpoint is a significant indicator for judging or assessing whether the toxicant or the contaminant is toxic or not. We saw the either LC50, IC50, EC50, LD50, and so on. So those points are the points that can kill or inhibit or minimize or retard 50% of the population of the exposed organism, either plant, microorganisms, animals, and the case may be. So any values we get on the calculation using the model, the linear regressional equation, that is statistical software, get it and then use it to establish the activity, which we can now apply in our curriculum, okay, and get our values, okay. So we want to say that if you enjoy our lecture series, you can subscribe, like, comment, and share. Thank you very much for listening, and stay blessed. Bye.